one more minute and then. Okay, so Antonio, if you agree, we start now. So hello to everyone. My name is Luciano Saso. I'm a professor of the Faculty of Pharmacy and Medicine of Sapienza University of Rome in Italy. It's a great pleasure to welcome you today at this webinar together with Professor Antonio Quadrado. This is the second webinar that we have on the Redox series that we started already last year together with some other colleagues from Italy, Alessandro Gioffre from the Italian Consiglio Nazionale delle Ricerche, the Italian CNR, Professor Mauro Maccarone from the University of L'Aquila, Marzia Resi and Elena Forte from Sapienza University of Rome, and Alessandro Leuti from the Campus Biomedico in Rome also. And this webinar is the second one that we organized in cooperation with the Coast Action CA 2121, entitled From the Bench to Bedside Transition for Pharmacological Regulation of NRF2 in Non-Communicable Diseases, coordinated by Professor Antonio Quadrado. And it's really a pleasure to cooperate with you, Antonio, for this series. The title of the webinar today, as you know, is NRF2 and Vascular Diseases. We have three distinguished speakers. We are going to listen to them. They will give their talks, and then we will open the discussion at the end. So I ask all participants to put the questions in the chat so that at the end we will have this final panel discussion. Let me also thank very much Brigitta Buttari, also cooperating with us and with Coast Action. And without further ado, I leave the floor to Antonio Quadrado, and then I will introduce the first speaker.
Thank you very much, Luciano. I think we are having a very successful series of webinars on redox biology. And I really want to thank all the people who are behind the screen, just listening and learning like we are all doing. We already had one webinar on cancer and NRF2. Today we have one on vascular diseases. Probably we will have another one by June, July on NRF2 and systems medicine. This is something that has to be still decided. And I think all I have to do is to enjoy all together these wonderful webinars that we are going to have today. And that's it. So thank you very much to everybody.
Thank you, Antonio. So now it's a pleasure to introduce the first speaker, Anna Grokot-Prejak. The last name is challenging to pronounce for me. I hope I did not do it so bad. And she's from the Department of Medical Biotechnology, Faculty of Biochemistry, Biophysics and Biotechnology of the Jagiellonian University in Krakow in Poland. She will give a talk on novel atypical functions of NRF2 and KEAP1 in endothelial cells. Anna is an assistant professor at the Department of Medical Biotechnology at the same faculty as I mentioned at the Jagiellonian University in Krakow. She studies the molecular mechanisms regulating the function of endothelial cells and blood vessels. Her team's research showed that KEAP1, GPDH, and the nitric oxide synthase form an enzymatic complex catalyzing protein S nitrosation. This process is deregulated in aged endothelial cells and blood vessels, which leads to massive protein aggregation. Currently, she tries to recognize the significance of NRF2 KEAP1 imbalance and the KEAP1 dependent loss of proteostasis in the function of blood vessels. So very interesting abstract. So it's a pleasure to leave you the floor, Anna.
Thank you very much. Can you see my slides in full screen?
Yes,
yeah.
yes, we can.
Okay. Thank you very much. So I would like to thank Luciano and Antonio for the kind invitation. And since I'm privileged to be the first to speak today, I would like to briefly remind you that NRF2 is a transcription factor which is repressed by KEAP1, which targets NRF2 to degradation. And in the presence of oxidative stress, de novo synthesized NRF2 translocates into nuclei where it transactivates the expression of cytoprotective genes. And today I would like to discuss with you our data that show that the activity of NRF2 reaches beyond genes transactivation and that KEAP1 is not only a repressor of NRF2. And we mostly work with endothelial cells which line the inner layer of blood vessels. We were looking at the angiogenic response of human aortic endothelial cells triggered by two factors, GDF15 and SDF1. And we observed that the angiogenic activity of those cells is wholly abrogated when NRF2 is not present. And in this case, NRF2 was silenced by siRNA. And this effect was associated with the impairment of cell migration and formation of podosomes. And podosomes are actin-rich structures which are used by several types of cells, including endothelial cells, for migration. And you may appreciate them. Here you can see them in this inset as those round spots. And they are formed in control cells. However, they can be hardly seen in NRF2 deficient cells. So the angiogenic response depends on NRF2. However, to our surprise, we observed that neither GDF15 nor SDF1 triggers binding of NRF2 to its consensus sequence. Moreover, we observed that none of them, sorry, but something is wrong with my pointer, none of them transactivates ARE-dependent gene expression. So we may say that angiogenic response depends on NRF2 protein, however, not on its transcriptional activity. And this effect was also confirmed in the endothelial cells overexpressing dominant negative NRF2. As you may see here, the angiogenic response to GDF15 and SDF1 is completely preserved in the cells. So we were considering several hypotheses that could explain this phenomenon that we observed. So the first one was that maybe another consensus sequence can be present for NRF2. However, this seems to be rather unlikely. We were considering that the microRNA regulation by NRF2, however, no specific pattern discriminating between the control and siNRF2 cells was observed. We considered also primersic senescence of NRF2 deficient cells, since the morphology of those cells indicated that this might be the case. And indeed, NRF2 deficient endothelial cells are senescent, and this phenotype is associated by the increase in P21 and P53 proteins, so the proteins characteristic for the senescent phenotype. And this phenotype is dependent on P53, since when we downregulated P53 in NRF2 deficient cells, we were able to rescue the normal phenotype of the cells. The proliferation was brought back. However, there was no change in angiogenic response of the cells. And we were considering also the interactions of NRF2 with many other proteins. However, we found nothing interesting. And so this was sort of like looking for a needle in a haystack. However, finally, this needle appeared to be quite big, since then we thought that actually always the darkest place is right under the candlestick, and therefore our final thought was that maybe lack of the interaction of NRF2 with KEAP1 blocks angiogenic response. So we observed that the level of KEAP1 protein is higher in NRF2 deficient cells. And when we knock down KEAP1 in NRF2 deficient cells, we fully rescued the angiogenic response of those cells and also podosome formation, which shows that indeed KEAP1 must be restrained by NRF2 to allow formation of podosomes, to allow migration of cells and angiogenesis. And this data and these conclusions were fully in line with our in vivo observations, since we saw no difference in vascularization in murine retina between wild type and NRF2 knockout animals. And if we consider the fact that N-terminal domains of NRF2 are present in this NRF2 knockout mice, and the most important for us was NA2, which is responsible for KEAP1 binding. And this was confirmed by us by usage of the anti-NRF2 antibodies, and also previously shown by Yamamoto group, but they use in this case anti-beta galactosidase antibodies. We may expect sequestration of KEAP1 by those N-terminal domains of NRF2 in these animals. So to sum it up, we may say that NRF2 tethers KEAP1 to permit podosome formation and cell migration as the final stage angiogenic response of endothelial cells. And this means that NRF2 and KEAP1 mutually repress each other. But to look for downstream effectors, we looked for the proteome profile in control and siNRF2 cells. And it turned out that many proteins regulating actin cytoskeleton are differentially expressed in those cells. And our attention was caught by ROGAP1, which is encoded by RGAP1 gene. And these ROGAP proteins are GTPases. So they block actin cytoskeletal reorganization and endothelial cell proliferation and migration. And it happens so that this pathway can be somehow bypassed or overcome by VEGF signaling. So if the involvement of ROGAP1 in angiogenic response impairment in NRF2 deficient cells was true, we should observe sprouts in NRF2 deficient endothelial cells stimulated with VEGF. And indeed, that was the case. You may see that the angiogenic response is fully preserved in NRF2 deficient cells stimulated with VEGF. So we had a look on the ROGAP most closely, and we saw that this is indeed increased in NRF2 deficient cells, and it drops when KEAP1 is knocked down. What was confirmed with the immunofluorescence and also with Western blotting. And we were able to restore the angiogenic response of the cells devoid of ROGAP1 and NRF2. However, it was only in part, let's say. So we may sum up that when KEAP1 is not restrained in endothelial cells by NRF2, this leads to the overexpression or the overabundance of ROGAP1 and probably some other similar proteins. And that leads to the inhibition of small GTPases. We have no podosome formation, no migration of endothelial cells, and therefore there is no angiogenesis. However, we may say that this model is sort of the artificial, and ask the question whether there is any relevance for this mechanism, for example, in patients. And it seems that it might be, because NRF2 protein dramatically decreases in endothelial cells isolated from aged patients. And it seems there is the crosstalk between NRF2 and aging in general, since on the other hand, NRF2 deficiency triggers premature senescence phenotype both in human endothelial cells and also in murine aorta. Why NRF2 declines with age, we don't know. However, and we are going to, we plan to check it, to investigate it. However, we wanted to look at the mechanism of NRF2 dependent premature senescence. So premature senescence can be triggered by oxidative stress. And in our case, NRF2 is not present. Therefore, we expect to have oxidative stress in the endothelial cells. And to our surprise, we observe no much difference in the expression of the major antioxidative genes in the aorta of NRF2 knockout animals. There was a significant increase in this level of the superoxide anion. However, we observed no lipid peroxidation, so no oxidative damage in those aortas. And here in this case, iron citrate served as a positive control. And very similar pattern was seen in human aortic endothelial cells devoid of NRF2. So only slight difference in the expression of antioxidative genes. Not that much difference in the superoxide anion level, no lipid peroxidation, and finally, there was no reversal of the senescence phenotype when we use TEMPO, which is the specific scavenger of the superoxides. So we may conclude that NRF2 deficient endothelial cells and blood vessels are protected from oxidative damage. And the question is how? And we were looking at endothelial cell function, and nitric oxide is a very important mediator both for the endothelial cells and for blood vessels. And we saw a sort of the gradient of NO. You may see here that inside the cells there was an increase in NO in NRF2 deficient cells, no difference on the membrane. Whereas outside we observed like the sort of the lower release of the nitric oxide intermediates. And moreover, we saw that the level of endothelial nitric oxide synthase is a bit lower in NRF2 deficient cells. However, it seems to be more active, and a dramatic increase of the inducible form of nitric oxide synthase was seen in NRF2 deficient cells. And premature senescence may also be triggered by protein S-nitrosation. And this is the process of protein modification. This is post-translational modification. It's a covalent attachment of the nitroso group to cysteines, to thiols. And this leads to the changes in protein activity, localization, stability, and so on. And so far, this process has been considered to be non-enzymatic. However, it is reversible. So there are two enzymes, thioredoxin reductase and GS-NOR, which may switch off S-nitrosation. Okay, and we had a look how S-nitrosation looks in our model endothelial cells, and we found profound S-nitrosation of proteins in NRF2 deficient human endothelial cells, and also very strong S-nitrosation in murine aorta. The aortas are isolated from NRF2 knockout animals. And this was associated with the decrease in the level of denitrosating, of the genes coding denitrosating enzymes, both in case of the human cells and the murine aortas. So the question is, what the functional relevance of S-nitrosation is in endothelial cells. So to address this question, we treated the cells with DTT, so dithiotrietol, which is the specific reductant of S-nitrosation. As you may appreciate here, the S-nitrosation was gone in cells treated with DTT, and it led to the profound lipid peroxidation, but only in NRF2 deficient cells, which finally led to cellular death. So looking for the mechanism, we did the mass spec analysis of S-nitrosated proteins from NRF2 deficient cells and control cells, and we found that one of those proteins was related to redox signaling. And this was NADPH oxidase 4, NOX4. So we knocked down NOX4 in NRF2 deficient cells, and lipid peroxidation was blocked, and we rescued those cells from apoptosis. So this shows that S-nitrosation of NOX4 protects NRF2 deficient endothelial cells from oxidative damage. But how SNO is created? So this time we had so that was our first first experiment to be done. So again, we had a look on a KIP, and here you may see detection of S-nitrosated protein using 3 different methods: biotin switch assay with the detection with the immunofluorescent detection, biotin switch assay with the Western blotting detection, and also the most specific detection of S-nitrosation, which is done with the hemiluminescent method. And you may see that knockdown of KIP1 in NRF2 deficient cells fully blocks protein S-nitrosation. However, it doesn't bring back the expression of denitrosating enzymes. So, and in case of GS-NOR, which is encoded by ADH5 gene, there is completely no difference between KIP1 knockdown cells comparing to control cells, whereas in case of thioredoxin reductase 1, we observe like some effect, however, not really strong one. So it seems that this effect here is not related to the reversal of the level of denitrosating enzymes. So we pulled down KIP1, and we found that it pulls down with transnitrosating proteins such as GAPDH and protein disulfide isomerase. So these proteins, transnitrosating proteins, can sort of move NO moiety from one protein to another. And we confirmed, in contrast to protein disulfide isomerase, GAPDH precipitates together with activated endothelial nitric oxide synthase with KIP1 in NRF2 deficient cells, which is not the case in control cells. And if we knock down either inducible nitric oxide synthase or endothelial one, or GAPDH or KIP1 in NRF2 deficient cells, S-nitrosation is either fully blocked or in part. So this shows that those 3 proteins, KIP1, GAPDH, transnitrosating protein, and nitric oxide synthase, form sort of the S-nitrosation enzymatic complex in cells which have low level of NRF2 protein. So the key points from these studies are that NRF2 represses KIP1, and this means that those two mutually inhibit each other. So not only KIP1 is a repressor of NRF2, however, at least in endothelial cells, KIP1 must be restrained by NRF2. Otherwise, it makes a lot of mess in endothelial cells and impairs their function. Moreover, KIP1 may form S-nitrosation enzymatic complex together with GAPDH and with nitric oxide synthesis. And the third message is that N-terminal domains of NRF2 are present in NRF2 knockout animals. So if your process that you investigate depends not only on the transcriptional activity of NRF2, but it may also depend on the interaction of N-terminal domains with other proteins, mostly with KIP1, this may affect your the interpretation of your data. And with this, I would like to acknowledge my friends, the people who are involved in the study, and thank you for your attention. And I will be very happy to take any questions. Thank you so much.
Perfect. Thank you very much, Anna. Very interesting talk. As I mentioned in the chat, we will open the discussion at the end. So I give the floor again to Antonio for the introduction of the other two speakers.
Thank you.
Well, thank you very much, Anna. Let's move now to the next talk. The next speaker is Professor Andreas Daiber. He studied chemistry at the University of Constance in 1997 and holds a PhD in biochemistry since 2000, since year 2000. He is full professor in molecular cardiology at the University Medical Center Mainz. He has published a very high number of papers, and more importantly, he has a very high age index, over 69. So I think he is really an expert regarding cardiovascular disease and redox biology. And Andreas, we are very privileged to have you here. So the ground is yours.
Thank you very much, Antonio, for the introduction, and thank you both, Antonio and Luciano, for the invitation to this interesting webinar. So you can see my slide, my first slide?
Yes, thank you.
Okay, wonderful. So I will talk about the possibility that the NRF2 KEAP1 axis could be used as a protective pathway or mitigation strategy against environmental risk factors induced cardiovascular disease and neuropsychiatric complications. Also, I will really largely concentrate here on the cardiovascular side. So as you can see here, the impact, or there is an impact of environmental risk factors on cardiovascular disease and mortality, as well as mental illness. On the left side, you can see that so the cerebral complications or neuronal complications we have recently or actually put together in a review that is in revision at the moment. And there is really clear evidence that noise exposure, especially from road traffic, railway or aircraft noise, induces mental illness. For example, depression, anxiety, annoyance, also can increase the rate of suicide, and also causes neurodevelopmental disorders. So for example, cognitive and memory impairment. And also there's a quite recent study that noise exposure can cause dementia and cognitive decline, which is of course a very important finding in our times, because dementia is a growing problem in our societies. And the odds ratio or risk for all of these complications is increased with higher noise exposure. And then there is also very strong evidence, especially coming, for example, from the World Health Organization environmental noise guidelines for the European region, where especially ischemic heart disease incidence, prevalence, and mortality was found to be increased with increasing noise exposure. So therefore, you can see here very nicely the estimated relative risk per 10 decibel increase in noise exposure, and that it's clearly above 1.0, so therefore it's increased. So keeping this in mind, that there is very strong epidemiological and clinical evidence for noise-mediated cardiovascular problems. So we should also keep in mind that noise exposure does not come alone. So because usually environmental risk factors, they tend to co-localize. And as you can see here, by showing the map of the United States of America, there is a co-localization of light pollution, air pollution, and noise pollution. So you see that especially in the highly urbanized and industrialized areas, there is a clear coincidence of these stressors. That's why in 2005, our colleague, Dr. Wild, has introduced the exposome concept. that is dedicated to study the impact of environmental exposures or risk factors on our health. And for example, chemical pollutants coming from the air, water, or earth, but also mental stressors or any other kind of stressor, physical like noise exposure, UV irradiation, they all can impact our intrinsic pathways and adversely affect, for example, the circadian clock, the stress response pathway by catecholamines and cortisol via the hypothalamic pituitary adrenal axis or the sympathetic nervous system. And this will ultimately then lead to inflammation and oxidative stress and end organ damage, which will then translate to health impacts such as stenosis of vessels that in the worst case will lead, for example, to myocardial infarction. So as mentioned, the circadian clock is a very prominent target of environmental risk factors. That's why we have recently summarized the redox regulation of the circadian clock. And as you can see here, there is multiple regulation sites for redox processes or oxidative stress. Here in the upper panel, you see the repressive pathways, and in the lower one, the activating pathways for the circadian clock, the clock B-mal1 complex formation. And part of them are related to direct redox modifications of cysteines in these circadian clock components, for example, in period or cryptochrome, that either prevents the proteasomal degradation or facilitates the proteasomal degradation. But there are also other players that are redox regulated, such as, for example, the AMP kinase or PARP1 or FOXO or PGC1 alpha, HIF1 alpha. And also I would like to highlight there is heme oxygenase 1 that can directly affect the binding capacity of BMAL1 to the clock component by binding of CO. So therefore, you see circadian clock is very important. And it's not only important because it's redox regulated, but circadian clock especially regulates a number of very fundamental processes in our body. And for example, it also regulates the stress response. And as you can see here, there is a clock regulation of the HPA axis that then circadianly regulates glucocorticoids, but also the glucocorticoid receptor activity is regulated in that way. There is also feedback then via period induction back to the circadian clock. And finally, of course, also stress responses show a circadian rhythm. Importantly, there is also vital interaction between NRF2 and the circadian system. And as you can see here, and of course is well known, that the NRF2 gene and protein itself is under circadian control and shows circadian rhythm. And of course, then also the downstream targets of NRF2, the already mentioned protective antioxidant proteins, they are under circadian control. But importantly, vice versa, NRF2 also has direct impact on the circadian clock, as can be seen here by data coming from wild type or NRF2 knockout mice. And you see that the NRF2 knockout causes a phase shift and amplitude shift in the period 2 activity or expression level. So therefore, NRF2 has a number of feedback mechanisms on the circadian clock via the nuclear receptors or the orphan receptors. but also directly on period cryptochrome complex formation and thereby inhibition of the clock. So therefore, important to know that NRF2 largely is regulated and regulates the circadian clock. So what we have summarized now actually in a review article is that different environmental stressors have a large impact, of course, on NRF2 activity, mainly by oxidative modification of the cysteines in KEAP1 or direct electrophile binding to KEAP1, which will then prevent the proteasomal degradation and allow the translocation of NRF2 to the nucleus. Of course, this can also directly be affected by other pathways such as histone acetylation or also DNA methylation that is also under control or modified by the environmental stressors. And then finally, in the nucleus, there will be expression of the known antioxidant response genes that are under control of NRF2. So therefore, it may be assumed that NRF2 is a kind of redox hub for environmental risk factors. And this also well in accordance with findings that we had in our studies because noise exposure increases the expression of hemoxygenase 1 mRNA, here in this case in lung endothelial cells, but also increases HO1 protein in the aorta of mice that were exposed to urban particulate matter, the form of air pollution. And also hemoxygenase 1 was increased in response to e-cigarette vaping in mice. And this was prevented by NOX2 genetic deletion as measured here by bilirubin plasma levels. So therefore the activity readout of hemoxygenase 1. So therefore we can say that probably all environmental stressors, they impact somehow the NRF2 and thereby also hemoxygenase 1 system. So I will only briefly summarize our findings so far that we had in our noise studies. What we did, we exposed mice with either around the clock aircraft noise, or we exposed the mice only during the sleep phase. What we observed was that there was a stress response. There was activation of the hypothalamic pituitary adrenal axis. And this was probably mediated by sleep disturbance, because we did not observe this when the mice were exposed during their awake time. And this went hand in hand with neuronal inflammation and oxidative stress, characterized by activation of the NOX2, but also, for example, downregulation of FOXO3 or neuronal NOS. And all of this led to adverse stress hormone signaling, so increased levels of corticosterone as well as catecholamines, and was associated with a systemic dysregulation of circadian clock genes, changes in key signaling pathways as shown by RNA sequencing that were pointing to dysregulated vascular function and remodeling, increased cell death, stress response, and inflammation. And this finally translated to functional impairment as envisaged by increased blood pressure, vascular oxidative stress, and inflammation and endothelial dysfunction that prime, of course, the organism for cardiometabolic disease. And our animal or mouse findings were well supported by a later study published by colleagues, Osborne et al. They found that subjects exposed to high noise levels, they have a higher amygdala activation, and the amygdala is a stress center, a flight stress center in the brain. And this was done by 18-fluor deoxyglucose PET imaging. And the amygdala activation was associated with increased atherosclerosis in the coronaries and increased inflammation in the coronaries. And all of this, the neuronal activation, the increased coronary atherosclerosis, translated to a lower MACE-free survival of the higher noise exposed group. So therefore, the higher noise exposed subjects had more major adverse cardiovascular events. So therefore, they had a worse cardiovascular outcome. So therefore, that was the basis, I would say, why we started the study, or we performed the study to mitigate noise stress and cardiovascular damage by NRF2 activation and hemoxygenase 1 induction. And the treatment regimen was as such that the mice received either 2 injections of hemine IP or 2 times gavage with dimethyl fumarate. And this was started on the day of noise exposure, on the first day of noise exposure, and was ended 1 day before the last day of noise exposure. And we observed that the treatments resulted in higher hemoxygenase 1 protein expression. That was also associated with higher levels of bilirubin as measured by HPLC in the plasma of the animals. And interestingly is also there is here no significant increase between control and noise only exposure group, which is probably due or is due to the ANOVA setup and the large data distribution here. So a t-test would have been significant. So therefore, we have a slight increase here in the hemoxygenase 1 protein, but we have a decrease in bilirubin, which indicated to us that there is probably, despite a higher level and probably activity of hemoxygenase 1 in the noise group, there is more oxidative breakdown of bilirubin, and therefore the levels are lower. So therefore, the concept was that noise exposure causes oxidative stress and inflammation, leading to endothelial dysfunction, hypertension, and therefore neuronal and cardiovascular disease. And this was also found. Noise increased the oxidative stress burden in the vascular wall, which was normalized or prevented by treatment with hemine or dimethyl fumarate. And we also observed the decrease of plasma interleukin 6 in response to the treatment. So therefore, there was normalization of inflammation. And also endothelial-dependent relaxation was normalized. So there was a significant endothelial dysfunction in the noise group that was prevented by hemine and dimethyl fumarate treatment, and also blood pressure was normalized. So therefore, in summary, we can say that dimethyl fumarate probably directly supported the transfer or translocation of NRF2 to the nucleus and thereby increased hemoxygenase 1 expression, among others, of course. And hemine probably via nuclear export of BAH1 also promoted this expression of hemoxygenase 1 and thereby increased the bilirubin levels, which is a strong antioxidant, the carbon monoxide levels, which acts vasodilatory, antithrombotic and anti-inflammatory, and also ferritin to decrease the free iron levels, which can explain the beneficial effects that we made under the treatments. So as a potential explanation for these beneficial effects of NRF2 activation on noise-mediated damage, we can summarize the following. Based on our previous studies, we know that noise key pathomechanisms are the activation or induction of the phagocytic NADPH oxidase, the NOX2, and this is mediated largely by activation of inflammatory cells such as lyse M positive monocytes. So therefore, we can say that noise exposure causes activation of the phagocytic NADPH oxidase and infiltration of monocytes, of inflammatory monocytes, because genetic deletion of NOX2 prevented the noise-mediated cardiovascular damage and complications, and also the ablation of lyse M positive monocytes by diphtheria toxin receptor overexpression on these lyse M plus cells and killing of these cells by diphtheria toxin treatment could prevent these effects. And going back to a previous study of our group, we were able to demonstrate that heme oxygenase 1 genetic deletion is highly protective, or is heme oxygenase 1 is highly protective against the phenotype, the pro-inflammatory phenotype of monocytes, and also phagocytic NADPH oxidase activity, because the gradual loss of heme oxygenase 1 activity in the heterozygote knockouts or the homozygote knockouts of heme oxygenase 1 was associated with a gradual loss in endothelial function, so therefore the worse endothelial function in the homozygotes. And this went hand in hand with the increased expression of NOX2 in the heterozygotes and the homozygotes, which was also associated with increasing vascular oxidative stress in the heterozygotes and the homozygotes. And this staining for reactive oxygen species could be omitted by PKC inhibitors or NOx inhibitors. We were also able to detect or to find that exercise is a very strong inducer of heme oxygenase 1, which is completely prevented by global AMPK knockout. And keeping in mind the publication by the group of Elke Heiss that AMPK phosphorylates the transcription factor NRF2 and thereby promotes the expression or activation of the downstream targets, such as heme oxygenase 1. This fits nicely together so that AMPK and NRF2 are also in a crosstalk. And this is also in line with our recent findings where we did really a huge effort in order to show that exercise, fasting, and ICAR, a pharmacological AMPK activator, are able to prevent blood pressure increase in response to noise. So all of these three treatments or therapies was able to prevent the blood pressure increase, prevented endothelial dysfunction in the thoracic aorta and also the mesenteric artery. And this protection was lost when we used endothelial-specific AMPK knockout mice, indicating that AMPK is really a central player here. And it would be wonderful to test whether NRF2 phosphorylation is also affected in either way by the protective measures or the AMPK knockout. So I would only like to point now briefly to a number of studies that are available showing that NRF2 is a wonderful mitigation strategy against air pollution. So there are really great studies using NRF2 knockout mice, for example, in diesel exhaust particles or also particulate matter with a diameter of 2.5 micrometer. And there are also a number of wonderful studies indicating that NRF2 is a very efficient mitigation measure against mental stress-mediated cardiovascular damage. And with this, I would like to summarize that we think that environmental stressors, they cause multiple disease And this can be air pollution, noise, UV irradiation, smoking, or pesticides or heavy metals. And they cause respiratory disease, metabolic disease, mental illness, cardiovascular disease, and skin damage. And all of this can be prevented or ameliorated, at least by NRF2 inducers or hemoxygenase 1 activators. And this, of course, gives a nice future outlook. So we have previously used the hemoxygenase 1 promoter activity assay using colon cancer hemoxygenase 1 promoter cells that co-express a luciferase construct, so therefore allow to trace the hemoxygenase 1 promoter activation by luciferase induced chemiluminescence. And of course, it would be very nice to use these cells for screening for environmental stressors and their potential. So this could be especially interesting for ultra or ultra fine particles, plastics, nanoplastics, microplastics, screening very quickly for different concentrations, chemical compositions or sources, and also, of course, e-cigarette or tobacco extracts or condensates. And at the moment, colleagues of mine in Mainz, they try to create an endothelial hemoxygenase 1 promoter cell line. And of course, it would be nice if here in the cost action, there are similar cells for NRF2 promoter activity. So despite the fact that all of these highly beneficial observations were made with the environmental stressors, and also a lot of preclinical studies were published on the beneficial effects of NRF2 activators for cardiovascular complications, there are dramatic setbacks, I would say. Of course, it's the general one that chronic therapy with NRF2 activators may promote the growth of cancer cells. There's a problem for any therapy where these NRF2 activators are used. But there is another one, this so-called Beacon trial. And there, patients with chronic kidney disease were treated with bardoxolone. And unfortunately, the trial was terminated earlier because there was a significant portion or rate of cardiovascular complications, mainly heart failure. So therefore, the outcome was not very beneficial. And the subsequent study speculated that this might be due to interaction of bardoxolone with endothelin receptor signaling, and this may be unfortunate for these patients especially. So therefore, as for cardiovascular use of NRF2 based therapy, we probably rely on the future studies, and at least I have found 2 of them that are obviously in preparation and will probably soon start recruiting. And one is the study on the impact of curcumin in patients with coronary artery disease. And the other one is a study to prevent doxorubicin-associated cardiotoxicity by sulforaphan. So therefore, these 2 studies will probably be quite interesting. And with this, I would like to thank you for your attention. And I hope I did not exceed the time too much. Thank you.
So thank you very much, Andreas. Antonio, if you agree, I saw a question in the chat from a person that is leaving now in a few minutes, Roberta Foresti from Paris. So maybe if Roberta is still there, maybe she can listen to the reply from Andreas. And she's asking if noise is associated with a premature senescence in the vasculature or other organs. So maybe if you want to give immediately a reply to Roberta before she leaves.
Mm-hmm. So this we did not explore in full detail so far, but we checked for some markers of senescence, such as the checkpoint enzymes. and they were increased. So therefore, there is evidence that senescence is increased in response to noise. But good point. This might be worth to be investigated in full detail in the future.
Thank you. So this was an exception because we said that the questions will come at the end, but we made this small exception. Thank you, Andreas. So please, Antonio, please continue with the next speaker.
Okay, so Andreas, thank you very much for this wonderful talk. A lot of very nice information and wonderful slides. And now I want to go to the next speaker, and I am really very privileged to have the opportunity to introduce Professor Giovanni Mann. Professor Giovanni Mann works in the vascular physiology, on vascular physiology in the British Heart Foundation Centre of Research Excellence at the King's College London. And he is the president of the Society for Free Radical Research and editor of Free Radicals in Biology and Medicine. His group is investigating NRF2 transcriptional activation of antioxidant defense genes in vascular endothelial and smooth muscle cells exposed to oxidative stress in diseases such as gestational diabetes and ischemic stroke. I think one of the most relevant findings, at least on the recent findings of Professor Giovanni Mann, is the relevance of oxygen levels in cell culture, which are really critical in order to get the right result compared to what is happening in the body. So Professor Giovanni, like I said, thank you very much for being here and for giving us this nice talk. The ground is yours.
Thank you very much, everybody, for inviting me and letting me participate. I'm going to try to make the screen large and turn on a laser pointer. Can you see all this right now? Excellent. So I think what I'm going to do in the next 25 minutes or so is highlight the importance of cell culture, but under oxygen levels that the human body or the tissues or cells see in vivo. And I'm going to use some examples without giving you the full story, and I'm illustrating the publications that we've had. But I wouldn't do any of this work without my group, who I'll acknowledge at the end, but more importantly, without funding from the British Heart Foundation, Heart Research UK, and these two companies, Baker Ruskin, who make the oxygen-regulated workstations, and BMG Labtech, who produce a Clariostar oxygen-regulated plate reader. Without those two bits of kit and their long R&D support, I would not be speaking to you today. And I also acknowledge being a member of BenBedFar, and I thank my host, Redox 2022. So in the first slide, what I've done is taken an illustration from Tom Keeley and my review in Physiological Reviews. It's available online, 2019. And what I'd like to share with you is that if we look at the lung and we breathe air up here, we're breathing somewhere around at sea level 21 or 20.9 kilopascals. And as the air passes into the lungs, through into the bronchioles, etc., we see that the peribronchial level of oxygen ranges somewhere around 5 kilopascals. If we look at the bronchi to alveoli, it sits somewhere around 12 to 15 kilopascals. So the immediate take-home message here is the cells in the lung do not see 21% oxygen, the exact oxygen that you use in all your cell culture experiments, and most of my career has used in vascular biology. This now illustrates the oxygen in kilopascals from air sits around 21. And as we pass down into the microvasculature, you can see that it can drop down quite significantly to around 3 or 4% in the smallest vessels. So this is the lung. I illustrate the heart as another example taken from Tom Keeley's in my review, and I'm going to draw your attention to the coronary artery where my cursor is circling. Sitting here in the coronary artery, one of the interests of my research group is, can we study coronary artery endothelial cells and smooth muscle cells? And they're seeing between 5 and 7 kilopascals oxygen. Anyone interested in the myocardium or the pulmonary vein or other areas in this review, any organ you would like, we've searched the entire literature. I think it's important to say that this value here is in the human. So that's just two examples I'll show from that review article. If we turn to cell culture and we plot on the x-axis the oxygen consumption in attomoles per cell per second, and we plot on the y-axis the pericellular oxygen level that different cell types would be exposed to in culture, if we set the oxygen at 5 kilopascals, this number here, the dotted line, and we're looking at HeLa cells, they will very much stay around 5 kilopascals. If we look at umbilical vein endothelial cells taken from the human umbilical cord at delivery, they're somewhere around 3.7 kilopascals. This is the take-home message of this slide. If you're working with cardiomyocytes, be they neonatal or adult cardiomyocytes, and these cardiomyocytes are being paced or contracting, they will be consuming oxygen dramatically, so that the pericellular oxygen level, if set at 5 in the incubator or workstation, may actually be as low as anoxic. So it depends on the cell type that you're looking at, its origin, from which tissue or organ it comes from, where you should determine the oxygen in the environment. Now, how do we do all this work? This is Tom Keeley and Salil Srivastava, who's now in the States. These are our workstations, and you can see here, this is the Baker-Ruskin workstation regulated by oxygen, nitrogen, and CO2. And you can have one side of the chamber at 5 kilopascals oxygen, which is physiological for most tissues, or you could set it to hypoxia, the adjacent station. And the cells are cultured within these workstations. They never leave the workstation. They never come out to a laminar flow hood where you're re-exposing them to hyperoxia. So we have our cells adapted long-term to defined oxygen levels. And you can see Tom with his arms inside this chamber, and there's a screen where we can actually see the cells. And if you look carefully, you can have the cells living in here. The drama about this is that the workstations are expensive, and if you have multi-users, you can have infections. And more importantly, if you have this workstations filled with cells or microtiter plates, it's very difficult to do microscopy and other things. And so for that reason, Baker-Ruskin have invented another unit here where you can isolate the cells under defined oxygen, seal them into this little condicell, and place them into a tri-gas incubator at defined oxygen. This is the other piece of kit that I wanted to show you. This is the Clariostar plate reader, which has injection ports and an oxygen control head or unit, so that you can take cells out of this incubator and transfer them very rapidly into this unit under the same oxygen and simulate ischemia reperfusion. That's just the kit. Okay, it's not letting me go forward. Why is that? Yes, it does. What we're also able to do is to use from Agilent a MitoExpress intraprobe. How do I get rid of this little here? And we can look at MitoExpress intra on the y-axis, the signal against oxygen on the x-axis. And as the oxygen level drops in the workstation or in the plate reader, as you can see here, we're getting a greater MitoExpress intra signal. So what we can then do in the readout is monitor the actual oxygen concentration inside a living cell, as well as in the medium simultaneously, under whatever ambient or pericellular oxygen level we set in the workstation. And this is illustrated here from umbilical vein endothelial cells, where the oxygen content plotted here from 0 to 25. In cells in the plate reader in room air, you can see the cytosol is the dark symbols. It's around, if it's room air, or around 21 kilopascals. Within the incubator, you measure a cytosolic oxygen level of 16 or so kilopascals, and the medium is the open squares. If we drop to 5% oxygen, note a very rapid decrease, so that after about an hour or so, you've achieved intracellular oxygen levels that reflect what is going on in vivo, at least for the umbilical vein. That sounds good so far. However, let me just put up all the data so we go faster. In vivo, if this is the umbilical vein, there's blood flow. And measurements have shown that the oxygen content in blood of the umbilical vein is around 3.7%. However, in cell culture, static monolayers, there's a gradient through the medium. So even if you set the workstation at 3.7%, this gradient means that the cell monolayer may be seeing a lower oxygen. And that's what I'm illustrating here in the right-hand lower graph. This is intracellular oxygen in air. You can see it's around 16, 17% oxygen. If we chose 3.7 as reported in vivo, we are actually have a lower intracellular oxygen than measured in vivo. And fortuitously, when we chose 5, we were able to recapitulate the exact oxygen concentration reported for the umbilical vein in vivo. So it's very important, even if you use an oxygen workstation, to know whether you're working with static monolayers or monolayers under shear stress and flow, and the organ or tissue they came from, and more importantly, their metabolic activity. The other take-home message that I'd like to leave with you is the following. If we lower oxygen, this is intracellular oxygen on the y-axis on the right-hand side, 0 to 20. If we lower that oxygen from hyperoxia, standard cell culture, to 5% oxygen, as you see here, about, immediately what you will see is an increase in HIF-1 alpha messenger RNA. That's the classical response of lowering oxygen. In the blue trace, over days now, from 0 to 8, I'm showing you the downstream targets of HIF remain elevated for 2 to 4 days. So simply changing oxygen acutely, you are having a cell that has HIF-responsive targets activated. We chose to keep our cells under a defined oxygen level up to around 5 to 6 days, and under these conditions, we have minimal influence from HIF-1 alpha stabilization. And that's shown in this slide here, work taken from Tom Keeley's paper in the FACEP journal in 2017. I'm plotting here in panel A, the representative immunoblot. This is HIF-1 alpha. 18, 10, 5, 3, and 1% oxygen for 5 days. You'll see that HIF is only getting stabilized at levels of oxygen below when we have this as pericellular externally in the workstation. But if we plot the oxygen content in the workstation, or the concentration to HIF-1 alpha, if we plot it against the intracellular oxygen level that we can measure, the light gray curve, we are only stabilizing HIF at levels of oxygen below 2%, which fits with 1 or 2% being hypoxia. So at this point, standard cell culture has nothing to do with normoxia. It is a misnomer in all papers. 2, physiological normoxia is dependent on the organ or the site of tissue that you're studying. It's not the same in the brain, the lung, the heart, the kidney. You have to be specific about what you're studying. And finally, we all accept that hypoxia is usually below 1% oxygen. That terminology is critical for what I'll share with you in the next few slides. This we've seen, I don't want to go through it, under normal conditions, this complex is ubiquitinated via the 26S proteasome. With electrophilic, nitrosative, or oxidative stress, you change the conformation of this structure between KEAP1 and NRF2, and endogenous synthesis of NRF2 leads to an accumulation in the cytosol and nucleus, and we turn on our genes. I want to forget about all of that down here. Another important concept. I've been asked many times, if you put cells in air for many, many passages and then stimulate them with an NRF2 inducer, this is an electrophile diethyl malate, we get a robust increase in HO1. If we put them 1 week in air, another week in air, and then trans them for 5 days to 5% oxygen, we're losing that response. If we go air, air, 5, and back to air, we regain the response. If we keep them in 5 all the way through, the cells are here, and so on. What this slide is telling you is that the induction of an NRF2 target gene, which is very responsive under hyperoxia, is attenuated when cells are under physiological normoxia. And this shows plasticity. So in other words, cells are not permanently changed. Why am I raising this issue? I've had comments from people working with cell lines, HEC293 cells or cancer cells, and they told me these cells have been immortalized and under hyperoxia for 30 years. I do not need to study them under physiological oxygen levels. Have you ever heard of a cancer cell in vivo seeing 18% oxygen? Never. So we have to think about the cancer cell core of the tumor and its periphery. And it's really important that both tumor cells and endothelial cells, as I show you here, these are primary HUVEC, show this plasticity. Okay. Okay, why won't it go? There we go. In this slide, I'm picking up the negative regulator of NRF2, or rather of heme oxygenase that you can see here, BAH1. And if I can draw your attention just to the white columns, this is in air under controlled basal conditions. This is 1 day at 5% oxygen, and this is 5 days. We're increasing BAH1 protein levels in the HUVEC, or if we look at these cells basally in human coronary artery endothelial cells, ignore the black columns, they're not relevant. So basically, putting cells at physiological oxygen levels that recapitulate the in vivo oxygen levels increases protein levels of BAH1, leading to the downregulation of heme oxygenase induced either by an electrophile called diethyl maleate or 4-hydroxynonenal. Now, what we did, okay, this is very odd. What we did here is we adapted cells, endothelial cells, HUVEC, to 5% oxygen for 5 days. We transfected them with scrambled siRNA or sRNA for BAH1. If you look here in these cells with scrambled RNA under 5% oxygen, this is basal, this is HO1. You don't see the induction of HO1. But if we silence BAH1 under physiological normoxia, we recover the induction of HO1. In other words, the elevated protein levels of BAH1 under physiological normoxia prevented this that you see here when cells are under the normal or normoxic oxygen levels. Does this pertain to all NRF2 proteins? No, it does not, because in this slide, again, we have cells, HUVEC, adapted to 5% oxygen, and we're looking at GCLM now. Here you can see that under scrambled conditions, we can induce GCLM even in low oxygen levels by diethyl maleate, and there's no difference whether we silence BAH1 or not. That in itself is saying the following. I can't get rid of this thing. Under hyperoxia, we will see that electrophiles, NO, sulforaphane, anything will cause nuclear translocation of NRF2 and the upregulation of our well-known NRF2 target genes. Under these conditions in hyperoxia, BAH1 protein levels are low and the inhibition, for example, of HO1 or NQO1 is minimal. If we adapt cells to 5% oxygen normoxia, we still have nuclear accumulation, but now the elevated BAH1 protein levels selectively downregulate these genes, whereas glutathione-related genes are insensitive to changes in oxygen. And we believe it's these genes, particularly the cysteine glutamate exchanger, system CET, that are regulating antioxidant defenses in vivo. If we move now very briefly just to brain endothelial cells, these are brain microvascular endothelial cells. You see again here in this panel on the left-hand side, HIF1 alpha stabilization only in cells adapted to 1% oxygen, whereas physiological normoxia, we have no stabilization. This is data from several experiments. Next that we see in panel F is that at lower oxygen or normoxia, levels of catalase are down. And finally, intracellular glutathione levels are down. Why? Because cells under physiological normoxia are not exposed to hyperoxic stress. This is just for the lecture that gave us on proliferation. Here we're looking days post-seeding, and you can see that cells actually grow more slowly in this cell type under 5% oxygen than under 18. I'm just going to pull this up because I'm aware of time. What we've done here is taken brain endothelial cells, lowered the oxygen from air to hypoxia, and then back to air. And we've loaded these cells with a luminescence probe to measure reoxygenation increase in radicals that you can see in the black curve. If we give catalase, we get a minor reduction, but if we give sod or PEG sod, this signal is completely taken out, as you see in these data. In other words, this burst of radicals that we also see in vivo in the brain is only occurring during reoxygenation. If we pre-treat the same cells with an NRF2 inducer, sulforaphane, and then run this experiment again, you see here under basal conditions or under conditions in the absence of sulforaphane, the radical burst, and sulforaphane activation of NRF2 genes, I can't show you, markedly lowers that response. So again, these experiments are conducted in that plate reader. And finally, if you take cells and you silence NRF2, which is the light gray curve, you greatly augment the radical burst. In other words, we feel in these cells, basal NRF2 is in fact mopping through its target enzymes, the radical burst, and protecting cells. Briefly, if I may, go to eNOS. We've heard about this. This is how I started my career with Salvador Moncada. We were interested in NO production and whether the production via endothelial NO synthase is affected by oxygen. And firstly, I plot to you on the y-axis here, data taken from Tom Keeley's PhD, intracellular cyclic GMP levels in umbilical vein endothelial cells that were cultured under 5% oxygen, the squares or circles open, 18 hyperoxia, and we are giving an NO donor. Note what you see immediately is that in hyperoxia, this is the dose response to DTNO, the NO donor. This is markedly augmented here when we have cells adapted to physiological normoxia. Simple take-home message here is NO bioavailability is enhanced when cells are not exposed to hyperoxia and free radicals. Here we use the proximity ligation assay. It's maybe not that visible to you, but we have cells in the upper panel under vehicle or histamine, acute stimulation, and we're looking for PLA and the proximity ligation assay. DAPI is the nucleus. You see a few red squares, which are PP2A and eNOS co-localization. It increases when we give histamine in air, but when we come to 5% oxygen, there's a marked increase in the association of PP2A and eNOS, and that shows you that the phosphorylation of eNOS, I couldn't show you that data, is markedly enhanced, the dephosphorylation, when we're at 5% oxygen. So when you do your NO measurements in air, the dephosphorylation of that enzyme, that's the phosphorylated eNOS, is delayed, but we can show that here. And these are work that we've just done with Emre Eroglu based in Sabancı University in Turkey. This is literally just accepted a few weeks ago. I'm illustrating here HEC293 cells in panel A, transfected with the genetically engineered NO probe. And you can see here the basal fluorescence in air, in HEC293, or in EAY HY926 cells. If we move to 5% oxygen for 5 days, because this genetically engineered probe, if it sees NO, it gets its fluorescence quenched. You can see the quenching of fluorescence in both basally, and that's what this is showing here. But more importantly, if we plot on the y-axis the change in NO sensitivity or signal, and we give an NO donor, as you see here in HEC293 cells, under 18% oxygen, hyperoxia, we get a slight signal. But if the cells are adapted to 5% oxygen, a much greater signal in the genome probe, both for these cells as well as for the cells that are endothelial origin. In other words, an NO donor, as I showed you before with cyclic GMP and HUVEC, has greater bioavailability if the cells are protected from hyperoxia radical generation. And finally, if I may end with this slide, these are EAYHY926 cells in air. There's another really important point for cell culture people looking at drug translation. We're looking at the NO signal on the y-axis, time on the x. The gray is a control cell, stimulate with ATP, which should turn on eNOS and make NO. If you add very acutely iron sulfate and ascorbate you markedly augment that signal, as you see here. What that's telling you is that all cell culture media that are culturing cells under hyperoxia are leading to the oxidation of iron, and the iron supply, which is required for this genetically engineered probe, is limited. But by supplementing the medium with iron sulfate and ascorbate acutely and removing it, you get a robust signal. These are data that we've done in London in the plate reader, again, cells loaded with the genetically engineered NO probe. Here we have cells at 18% with no iron or ascorbate, small signal. If we add iron sulfate and ascorbate, we enhance the signal and we can take it out with the inhibitor of enos L-name. But if we put cells at 5% oxygen for 5 days, the signal is enhanced compared to here. And now look at what happens when you add iron sulfate, very low concentrations acutely, you markedly augment that signal, which is abolished by L-name. The take-home message here is NO bioavailability in culture, in cells, is enhanced if the cells are maintained under physiological normoxia. 2, anyone working with cell culture, please read this paper because it has lots of supplementary data on the importance of providing iron sulfate and ascorbate, because not only will it affect the GNOP probe, but it'll affect all metalloenzymes and proteins. 2 more slides, if you bear with me. This is viability, the most boring assay on earth, against ionomycin to load calcium into a cell. These are cells in the open circles looking at death of cells in air, but if we adapt cells to 5% oxygen, we protect. In other words, we are protecting against calcium overload. If you don't buy that, which is an MTT assay, we're looking at an exon 5, that's the phosphatidylserine groups exposed, big reaction, very little annexin 5 staining. This is the damage done when you give ionomycin in air, the white columns, in black, markedly protective. And then because I talked about the lung at the beginning, all I want to share with you here in this paper is the first column here on this side are cells cultured in hyperoxia, standard culture. Glutathione levels sit at around 45 nanomoles per milligram protein. If I take the same cells and I move them across here to the other workstation and I maintain them at 13% oxygen, like in vivo, glutathione levels are virtually halved. Again, an index that cells are high glutathione because of the continuous activation of NRF2 and the targets such as XCT and GCLM, etc., that lead to glutathione synthesis. The other take-home message here is we're testing nanoparticles to see if they're toxic. In air, these cells are like concrete. Only high concentrations kill. Here, let's go to the clinic. We're ready to roll with patients. This side over here, look at the nanoparticles killing off the cells because the cells are under physiological normoxia. That's our physiological lab. I can share that with you, etc. But I thought this would be a fun movie because we heard earlier, can I make it work? How do I make it? How can I turn this on? I can't. Do you know how to make this movie work? I don't. This here, if I can't do it. Maybe if I end the show, it won't work. Here we go. Can you see that on the screen at the moment?
Yes.
Yes.
So this is what we're doing now. We're interested in human coronary artery endothelial cell migration, and we're doing these experiments under defined oxygen. So hyperoxia all the way down to hypoxia. And the take-home message here is, although I haven't said it to you without showing you the data, is that the migration is far better under physiological normoxia for this cell type. So it's cell type specific. And finally, as I promised, I'll go back to full screen. These are the guys who I owe my thanks to. I couldn't have done it without any of them. Here's Tom Keeley, a big player, Sarah Chappell, Richard Xiao. Here's Fan Yang, who works in smooth muscle cells. Gabby did all the brain work. And as I said, without them, I couldn't do anything. Thank you for your time. I should stop sharing somehow. How do I stop? There we go.
So, Antonio, please.
Okay, Giovanni, really thank you very much for this very nice talk. Very, very informative, very illustrative of the situation with normoxia and all the mistakes that we make working in hyperoxia. And you know, nobody is willing to change, and this is really a pity. So, okay, this is what we have. So maybe Luciano, what we can do now is to start a discussion. Can you go over the chat first, please?
Yes, so first of all, many thanks from me as well to the three speakers, really excellent talks, very interesting. Also, I want to thank the participants who are putting questions in the chat. We have a very active participants. Anna has been very efficient, so she replied already to some of the questions that were addressed to her. But of course, Anna, if you want to add any comment, please take the floor. Otherwise, we can move also to some questions that were addressed to Andreas. I don't know, Anna, if you have any additional comment on the question that were addressed to you.
Well, I was trying to address all the questions that appeared in the chat, and well, if there are more questions, I will be happy to answer.
Thank you. Yeah, you have been, as I said, you've been very, very efficient. So, Andreas, as you probably noticed, there were some questions for you in the chat. One was from Roberto Motterlini, Université de Paris Est in France, and is asking, are you trying to dissect the specific role of CO and bilirubin in their ability to reverse noise-mediated cardiovascular dysfunction? The effect of noise on blood pressure is quite remarkable. Do you think CO contributes to this effect, or is bilirubin equally important?
Because at the moment I have no answer to that. So we did not discriminate between the two. So this could be maybe done by the CORMs, by the CO releasing molecules that Roberto also developed, and bilirubin, yeah, could be probably also treated. So therefore, there would be a chance to see which of these two metabolites may be more important.
Thank you very much. There were a couple of questions also for Professor Mann. One is it known why the BAC1 levels are high at low oxygen levels? Is the increase due to enhanced transcription or increased protein stability?
Albina, it's a very good question. I don't have the answer for it. I wish I did at this stage, but we don't have that yet. There is evidence also, again, cell type specific, that BAC1 protein levels are elevated in lower oxygen. The word lower oxygen frightens me, because when you say low oxygen, one always associates that with hypoxia. And I would like to get across the concept that if you lower oxygen from air to whatever you lower it, you're going to turn on HIF1 alpha. But that's not hypoxia. That's just lowering the oxygen response. If you're going to do experiments, and I really, Antonio, I'm going to take you up on your comment. People don't want to do it because it's expensive and a pain in the backside. My answer to you is, if you're designing NRF2 target drugs or inhibitors, I would strongly advise you do this in cells that are as close to possible to in vivo situation. If you're working with animal models, goodbye to my cell work, but we can't really get at all the molecular mechanisms. So it'll be a long answer, but no answer. I don't know if it's either of the other. And there's a question from Roberto Montellini. Very good question. Did you review the paper? I don't know. But we were asked very seriously, if we give an NO donor, be it NOx7 or DTA-NO or SMP, all have their problems, are we delivering more NO to the cell in higher oxygen than in lower oxygen, right? In other words, if I read your question correctly, I think that adding in 18% will have more nitrite being generated than in lower cells. We have not looked at nitrite or nitrate or tried to measure it, but I come back, as I tried with the referee's comment, is when we stimulate endogenous NO from enos by using ATP as an agonist in endothelial cells, we see far greater sensitivity or signal if cells are in 5% oxygen. And that's because in hyperoxia, oxygen is scavenging NO, right? And so if we're trying to understand how a cell is responding to an agonist or to an NO donor, contemplate what it would be like if the cell cytosol sees 16% oxygen compared to 3.7. What are the mitochondria doing? What's the ER doing? What's transcriptional machinery doing? So I think it's important. And then there was one more question, I think. No, sorry, Roberto, I wish you had reviewed it. I'm just kidding. But the work that we're doing with EMRA, who I really should say is our champion of these genetically engineered probes, who study with Tom Michel in Harvard, this has proved to be a fantastic collaboration. And my colleagues Fan Yang and Matt Smith have been the people doing the work here in London using that. And we've tried to have our workstations and a plate reader to do the experiments. So read the paper when it comes out. It's fun.
Thank you. Thank you very much, Giovanni. Also, thank you to Roberto for being a very active participant. There is another question for Andreas from Gunay Yatik Anachak from Turkey. Is there a crosstalk between HO1 and the hydrogen sulfide? Have you checked the effect of noise on H2S?
There is this crosstalk between the hemoxynase 1 and H2S producing enzymes. It would be quite interesting. We don't have the tools for that. So of course we could check for the expression of the enzymes, the CSE or something, but we don't have the tools to monitor, for example, H2S formation.
Thank you, Andreas. So these are the questions from the chat, but of course now we would like to open discussion with the panel. I don't know if Antonio, you have questions for our three excellent speakers.
Thank you, Luciano. I have a couple of questions, if you mind. These are for Anna. Anna, you found this very interesting protein complex with GAPDH and INOS and with KIP1. And my question is if you can find in this specific kind of complex, if you can also find NRF2. I understand that the way to do it probably is to inhibit KIP1, probably with the NET8 inhibitor. But my question is whether you could also co-precipitate NRF2 under these conditions.
Well, so in these conditions, NRF2 was not present because it was done in NRF2 deficient cells with knockdown of NRF2 with siRNA. So, you know, we haven't tried to look at NRF2. However, if we block KIP1 in the cells, well, I don't know what would happen.
I understand that you didn't. I mean, my question is in normal cells, whether not in the NRF2 knockout cells, which I know that.
Oh no! So yeah, okay. So so the complex is not formed then, because yes, because we had the control with the normal cells, and where NRF2 was present, and the complex was not formed, and there is no nitrosation of proteins then.
Okay, and the second question, which is, you know, GAPDH is a very good substrate for autophagy, for chaperone-mediated autophagy. And we know that P62 is a carrier protein for KIP1 to autophagy. So my question is whether there would be a parallel idea with GAPDH taking KIP1 to chaperone-mediated autophagy.
Well, could happen. However, in the cells with NRF2 deficiency, P62 is decreased and autophagy is impaired. And therefore, we believe that KEAP1 cannot be destroyed, and therefore it increases, the level of KEAP1 increases in those cells, and therefore it may form the complex. But well, maybe you're right that there is a place for GAPTH in the KEAP1 autophagy degradation.
Thank you. If I may, I will finish with another question, in this time for Giovanni. So Giovanni, it seems to me that when we use any NRF2 activator in cells, we get very robust data. When we go to animals, the data are not so good. We always claim that the reason is pharmacokinetics, but maybe there is another reason according to your data. Maybe just the animals are not as responsive as cells. What do you think?
I think, Antonio, I tried to illustrate that point with two experiments. When you adapt cells to physiological normoxia, i.e. 5% endothelial cells, any NRF2 inducer, sulforaphane, NO donors, hydroxynonenal, what have you, will not lead to a robust increase in NRF2 target genes. You don't see it in immunoblot. Forget about the messenger RNA, just the protein. And the likelihood is that for some reason, that hyperoxic environment in which the cells are kept in standard cell culture is facilitating that response. In the same way that tumor cells have that upregulation in the same way. When you come down to 5% oxygen, what we noted is that the NRF2 target genes, GCLM, GCLC, and XCT, are not affected by oxygen. And we thus postulated in one of our papers that we believe these genes or these proteins are the ones responding acutely to oxidative stress in vivo, but not HO1, not NQO1. Do you see what I mean? Now, I know NRF2 regulates over 200 different targets, but it's possible that the milieu of oxygen radicals in the blood vessel wall or in the heart under normal health conditions isn't that. So when you try to screen an activator in vivo in mice or humans, it might be very different. And that's why I say, look at the molecular mechanisms, or at least the image of that pathway in a cell as closely adapted to physiology. That was the rationale for this.
Thank you.
Can I share something crazy with all of you? Because I'm crazy, as you all know. I have a group from France, from the Marine Laboratory in Brest, who work with oysters, right? And you know, oysters sit in the sea and they filter the water when the tide is in. They eat and they're oxygenated and their pH is good. When the tide rolls out, the oysters slam shut tight. The oxygen drops to anoxia for 6 hours. The pH, you would fry, we would die. As soon as the tide comes in again, that oyster opens and it literally has ischemia reperfusion injury going on every day, every 6 hours. That oyster is coming to Giovanni's oxygen laboratory to isolate hemolymph and smooth muscle cells from the valve to study them under our defined oxygens. I asked, could I have a special box of oysters that I could eat? But they smiled. So there are really important things that are going on in nature that are giving us insights into what real oxygen levels should be versus what we do when we do cell culture in air.
Thank you, Giovanni. I hope you invite me to your laboratory to have
exactly.
So I think Anna has a question.
Yes, thank you. I have a question to Giovanni. Thank you for your excellent talk and for your important insights on the oxygen level in our cell culture. Another issue is the lack of laminar flow or any kind of flow and shear stress. Have you tried to perform any experiments in lower level of oxygen and with shear stress?
If I'm honest with you, we've done pilot experiments, and that's the next piece of work we're doing. We're taking our endothelial cells in static monolayers and culturing them in IBIDI flow chambers, but within our workstation. It's a pain in the backside because of the tubing, etc. But we're set up to do it, and we're interested in what shear will do to basal NRF2 levels, obviously, and enos, which we're doing. And we're doing that in coronary and in brain microvascular endothelial cells. So watch this space. I still have energy.
Okay. And another one, if I can have, because if I remember well, there was a paper from Shibahara group published in 2003, I believe, and they showed that both hypoxia and cobalt chloride, which mimics, let's say, hypoxia, decrease the level of BAH1.
Yeah. So if we have followed in some cell types BAH1 at 1%, but the real emphasis that we're interested in, largely driven by the reviewers, they said, well, look, guys, when you can't induce NRF2 in a cell type, that cell type must be dead. It's not working. And that's where we had to come back and say, look, we've run a full scale of the oxygen levels, and we've shown you that HIF stabilization is only occurring at 1% and not at 5%. The fact that the cells weren't upregulating NRF2 targets was a consequence of the oxygen environment and the fact that the cells didn't see hyperoxia. Yeah, that's why in your cells, I ask you, when you have your normal cells in air, yeah, with lots of hyperoxic stress, and you now take away NRF2, maybe that small change in oxidative stress you don't see as more radical generation. Do you see what I mean?
Right. Yeah, right. But the point is that we try to isolate endothelial cells from NRF2 deficient animals. They won't survive in normoxic conditions, and they can survive only when we put those cells immediately to hypoxia chambers, right? And then they will.
That's one
otherwise
thing.
It won't work.
The other point is, if you call them hypoxia chambers, that's also not a good thing. Call them oxygen regulated.
Okay.
Physioxia. There's a clue with endothelial cells to isolate or smooth muscle cells from NRF2 knockout animals. If you're doing it in room air, put in a tiny bit of mercaptoethanol, because what that does is elevate the glutathione, and then the cells will proliferate. But if you take them out from an NRF2 knockout and you try to culture them, we were doing that with aortic smooth muscle cells back in 2004, you needed mercaptoethanol to get them going. Yeah. One
more question, if I may have. Do you think that the effect of hypoxia or lower oxygen level on BAH1 expression can be cell type dependent?
Yes. So that's the other thing that I think is important. I'm about to put together, if I have enough strength, a crazy paper of the effects of physiological normoxia on multiple cell types, all the things we've measured. So it becomes like a Bible, because people think that if I lower oxygen, this happens everywhere. It doesn't. And a smooth muscle cell, for example, coronary artery smooth muscle cell, I didn't show you data. If you stimulate it with hydroxynonenal or sulforaphane, it doesn't care about the oxygen. It turns on hemoxygenase. I don't know why, but that cell type comes from a different source in that coronary artery. Do you see what I mean? So you have to be specific in what you're doing.
Right.
Yeah.
Thank you.
You're welcome.
I think Andreas has the raise his hand.
Yes, thank you. I would have a question to Anna. So this was really substantial nitrization that you showed. So, and I wonder whether the cells, they liked it. So do they really feel well, the cells, or because this high nitrization levels should rather create a lot of stress on the cells, especially as the high NO levels or nitrization levels, they interfere with energy metabolism quite a lot. So we know many thiol-dependent enzymes in the Krebs cycle, for example, isocitrate dehydrogenase, GAPDH, many more, so that are inhibited by S-nitrization. So therefore, how do the cells cope with that?
Right. So for endothelial cells, it seems devoid of NRF2. It seems that S-nitrization is necessary for the cells to survive. And you ask whether they are happy with that. Well, this is the let's say, the choice of being either alive but senescent, or to undergo apoptosis, because what we observed is strong S-nitrosation. It protects the cells from apoptosis in this case. However, it leads to the senescent phenotype. And all this stuff depends on the level of KEAP1 and on the levels of NOx4. So if you have a choice either to live but to be prematurely senescent or to die, you know, this is the matter of the level of S-nitrosation on proteins.
Okay, this I see. The other point is we know that NO itself is not a so good nitrosating agent. So therefore, usually you need, let's say, oxidative nitrosation called in order to really make this widespread nitrosation that you have shown. The question is, would you find less nitrosation if you would work at a low oxygen tension, as Giovanni says?
Well, we haven't tried, but probably this might be the case. And what we believe happens in the cells is the overproduction of the superoxide anion may facilitate S-nitrosation, because, as you said, for this reaction, NO is not, let's say, the presence of NO is not sufficient. However, if it is in the close proximity of the superoxide anion, which may happen due to the inos uncoupling or the production from NOx4 or NOx2, well, this would create the environment for the S-nitrosation to occur. However, whether it happens in this way, I don't know. Another idea was that the zinc anion is replaced with the Fe3+ in KEAP1, and maybe in this way it would facilitate the formation of S-nitrosation on the proteins. However, we haven't expected it. I mean, we haven't examined it, and well, this is something that that must be done simply to elucidate how the reaction itself goes on and how it occurs in the cells.
I don't know. I can't see myself. Can I ask a question?
Yes, please.
It's Giovanni. I just wanted to ask two things. Firstly, say something. When cells are in hyperoxia, air, total or large ferritin levels in the cell are increased. So labile iron is limited because it's binding labile iron. That's point number one, really important. The second thing is, when you're in hyperoxia, the iron in your culture media, whatever you're using, is oxidized. And the only iron that's transported is Fe2. So you're going to be limiting the iron delivery to your cells. But what I was going to say now to Anna, or more as a comment, if your experiments were conducted in physiological normoxia and NO bioavailability was enhanced, which I showed in the data I showed for endothelial cells, if I understood you correctly in your lecture, you were suggesting that the large amount of NO or nitrosylation was coming from inducible nitric oxide synthase. Is that right?
Yes, this is right, because let's say the most pronounced effect when we wanted to block S-nitrosation was achieved when we blocked inos, in contrast to inos.
But why would what I'm asking is, why would your endothelial cells, so I'm just taking a photo of all of us, why would your endothelial cells express inos? Endothelial cells very rarely express inos, even if you expose them to lipopolysaccharide, TNF-alpha, what have you. But you seem to have endothelial cells that express inos.
Well, this is not very surprising for us, because as we checked, when those cells, these are human aortic endothelial cells, the primary cells, which are commercially available. So, and we tested them with the specific antibodies recognizing inos, and we confirmed let's say, the specificity of those antibodies with siRNA against INOS. So we believe that this is the specific signal for INOS. And we observed also that IL-6 or soluble ICAM or TNF is increased. So general, there is sort of the inflammatory phenotype in those cells. So
yeah, I mean, I buy this.
You know, this is not surprising that INOS is also there.
No, but what I was going to say is I don't in any way contend. I believe that your endothelial cells in air are under an inflammatory milieu, and so everything will be upregulated. But if you read the endothelial literature, right? I'm not saying you haven't, please forgive me if I'm not sounding polite. I mean to be polite. I am not aware that INOS plays a significant role in endothelial cell function. INOS comes from infiltrating macrophages when they move into the plaque. INOS comes from neutrophils. INOS comes from inducible cells. It's an inducible enzyme. It's not a constitutive enzyme in endothelial cells. That's all I was trying to say. And I feel that if you move it to lower oxygen, you will have less of that INOS contributing, and maybe, as you said, less nitrosylation, as you showed.
Okay, so we must try. Okay, so
yeah,
so
I think it's a good idea to try.
Yeah. Okay. Thank you.
Thank you very much to everyone. Really, it's a very lively discussion. Many good questions and comments. I see again Roberto Mortellini also with some other interesting comments. One is related to NO and the O2 levels. And this says that they have data showing much less INOS expression and nitrate production in macrophages challenged with LPS at 5% O2 compared to 20% O2. This was one question from Roberto. By the way, he's based in Paris, so he may be also expert about oysters. Maybe useful.
Yes.
And another comment also from him, he says, if we want to develop future drugs to be used in vivo and need to screen hundreds of compounds in vitro, 18-20% O2 must be avoided. Not only O2 has to be lowered, but the O2 levels must be kept approaching zero, which is the situation in vivo. So these were comments. I don't know, Giovanni, if you want to add anything.
No, I mean, look, it costs money to do everything, right? And money is short and life is difficult. But I think the failure of labs to adopt this strategy, I'm not novel in this. My lab isn't novel. This has been going on for years. People just don't want to do it. Why would you want to put your arms in? You know, and all the people that work with stem cell work, they know about this. Stem cells proliferate at 3% oxygen beautifully. And if you engraft them into a myocardial infarct region in a mouse heart, they repair. But if you culture the stem cells in air, it's not as good. So there is a real precedent for doing this. And I think a lot of the clinical translation that hasn't taken place is because we've been exploring the biology, if you wish, and the signaling under hyperoxia. And that's my contention. And I wish I could have a paradigm shift. I mean, I'm being honest. I've had discussions with colleagues who said, as I said at the beginning of my lecture, my cells have been in hyperoxia for 30 years. I'm not going to change. And the answer I gave, which wasn't a nice one, I said, don't let your grants come across my desk. I joke. But I think, look, we have to be practical. We can't say everyone has to do this because I gave a lecture. We have to be sensible. But I think in the NRF2 consortium that we have, Antonio, there is scope for at least doing something to see whether there is a difference or not. And even if we only found out that glutathione targets of NRF2 are the ones working in vivo, that's pretty good information for targeting drugs. Do you see what I mean? That's my view. Sorry.
Okay, so maybe I have, Luciano, I have received a couple of emails asking me if these webinars are being recorded, how we can access them.
On the same website, antiox.it, you can come back to the same website, and then in a few days you will see the recording, exactly like you can see the recording of the previous webinar that we did. So just come back on the same website, antiox.it.
There is another question. Please, yes. Alexandro.
Can I? So congratulations for the excellent talks to the three panelists. I had a question to Giovanni Mann regarding the H2S signaling. So I was wondering whether he is planning to study the H2S signaling under physiological conditions, or if he has already data on that.
The honest answer is, I have never looked at H2S signaling, as interesting as it is. I think it's fascinating, and it would be fantastic to review it under conditions in which cells have been adapted to physiological oxygen. I think the word adapted is important too. If you do this acute transfer to low oxygen, that's not the way to do it. You've got to set the redox phenotype. And I am certain that H2S signaling will be different.
Absolutely, and actually that would be extremely interesting because there is a very interesting interplay between oxygen H2S, as you probably know, and also regarding the H2S consumption. So the way to keep H2S and fix bioavailability of H2S in the cells. So that would be extremely interesting.
I suggest to you, Alessandro, if you have a talented somebody and they wanted to come across with a defined question and cells that you well know, you're welcome to come here. You know, I can't pay for your flights or things, but I can cover the consumables whilst here, and you can do a test. And if you see differences, fantastic. If you don't, at least you can go away and say, I can continue in air.
Excellent. Thank you very much. Thank you.
Thank you. I see that Andreas was very kind to reply in the chat to another question from Brigitta, because we really need to close. I don't know, Roberto, if you have a very quick question. We have only 3 minutes left. If you have something super quick, please open the microphone. Yes. Thank you for being so active, Roberto. We don't hear you. Your microphone is not working. Yeah, because
can you hear me?
Yes, now we can.
Okay, sorry. Yes, I enjoy very much all the three lectures, of course, and I would like actually to say something actually in defense of carbon monoxide, because actually everybody's talking about oxygen, nitric oxide, and hydrogen sulfide. When we work at low oxygen levels, and when we work actually at physiological oxygen level, also the amount of carbon monoxide that you need actually to have a physiological effect is much, much lower. So the toxicity of carbon monoxide as well is not so much strong as you do when you culture actually the cells at 20% oxygen, because you need so much carbon monoxide. And then you know that the affinity of CO for heme-dependent proteins is much, much higher compared to oxygen. So we are actually studying the effect of carbon monoxide at low oxygen. And I think actually it's very interesting to see how carbon monoxide is not so toxic actually as many people claim, you know. So that's in defense of my, little friend carbon monoxide that I'm studying since a long time.
Thank you very much, Roberto. We have 1 minute left, so we need to close. Please, I give the floor to Antonio again.
Thank you very much to the speakers, to the participants and the audience. And let's go for another wonderful webinar in a couple of months.
Thank you very much, Antonio. Many thanks to the three speakers, really excellent. Many thanks to the very active participants, and again to my colleagues, Alessandro Giuffre and Mauro Maccarone and all the others. And looking forward to seeing you again in the next webinar of this series. We don't have a definite date yet, but we are planning to do that at the end of June. So please stay tuned, and we will announce on the website antiox.it the next webinar. So again, many thanks to all, and see you soon. All the best.
Bye-bye. Thank you.
Bye-bye. Thanks.
Thank you. Bye-bye.
Thank you.